So the mitochondria, as you guys, might remember from growing up and learning about biology, it's always known as the powerhouse of the cell. Why? Because it's the area where ATP is created. And we will go into, cellular respiration more in depth in a biochemistry lecture a little bit down the line. But for now, we're just, keeping with the mitochondria is responsible for producing ATP in the cell in the bodies, and so that the functions, functions can utilize the ATP energy to do whatever it has to do. But the mitochondria is also well known to be associated with the endosymbiotic theory, in which the theory was that the mitochondria were once prokaryotic cells that ended up becoming engulfed by larger cells and having this, symbiotic relationship. And why this was the theory was because mitochondria are special in the case that they have their own genome. It's this like circular genome, so that kind of, indicates possibly a prokaryotic cell. It also has the ability to self-replicate.
Really?
It owns ribosomal RNA, eRNA, proteins in the electron transport chain, and also of ATP synthase, which is used in cellular respiration. And also, it uses a different system of transcription and translation compared to the nuclear genes, which they use from the nucleus, and they have a unique genetic code on their own. But yeah, so this is what kind of indicates that mitochondria were once prokaryotic cells, but really because they are currently in the larger eukaryotic cell, a lot of the proteins that the mitochondria actually uses are actually from the nuclear genes, even though they do have their own system. It's just more, I guess, it's more energy efficient to just use the resources of the nucleus that's already creating these genes for the rest of the other organelles. So they're just going along with it. Yeah, and the mitochondria have the outer membrane and intermembrane space, and also an inner membrane. Yeah, and each of the different parts are 4 different parts of cellular respiration, which we'll go on a little more later in another lecture. And so now I kind of wanted to end with the last structure, a very heavily focused on structure, the cellular membrane. And so the cellular membrane, as you can see in this diagram, it's a semi-permeable phospholipid bilayer, and the phospholipid bilayer is made up of the phosphate head groups, as you can see in the red circles. Also a glycerol backbone, which is connecting these 2 fatty acid tails. And they have 2 of them, and they're kind of faced with the hydrophilic heads, which are hydro, water, philic, loving. And so they face the outside portion and then they also have the hydrophobic tails, which are the fatty acid areas, which are hydro, so water, and phobic, fearing, which are kind of stuck in the interior of this bilayer. And so they're also known as being amphipathic. So, having this water loving and having this water fearing aspect of it. And then also that, the semi-permeable phospholipid bilayer also regulates the things that go in and out of the cell, which is, very important, because if this is a cellular membrane, you really want to regulate the things going into the cell. But usually small and nonpolar molecules are the fastest ones to go through. The different ones, like larger molecules, tend to be, a lot slower, and especially when they're polar in charge. So these are all different factors that regulate that, but there are also different signaling molecules that help with transporting some of these other molecules into the cell. But the cellular membrane also contains cholesterol. So the yellow hexagons here in this diagram, I'm circling with my mouse. Cholesterol is really important for really maintaining, stability of the phospholipid bilayer, because in colder climates, when these layers will kind of try to harden, the cholesterol actually helps kind of make it a little bit more fluid when in cold environments. And then the cholesterol, on the other hand, in warm environments, when everything is a lot more fluid, it is able to create a more like rigid structure with this, surrounding fluid area. And overall, it creates this, overall, it creates this stability for the cellular membrane. It also contains different proteins. So there are integral proteins, transmembrane proteins, and peripheral proteins. So integral and transmembrane are usually like within the bilayer. So this is the integral, and transmembrane is kind of like similar looking. And then peripheral are usually on just like the outer portions near the hydrophilic head areas. And then also it has carbohydrates that are usually attached to a protein. So here you can see like the glycoprotein, or here you can actually see it just says carbohydrate, but these are the little blue dots coming out from the original protein. Yeah. And this is also a topic or a question that they like to ask in the MCAT at least once, or they like to kind of have it in a passage, and they mention, that a cell has these particular structures, which kind of denotes it as being a eukaryotic cell versus a prokaryotic cell. So I really wanted to stress this topic as well. And so what are the differences between eukaryotes and prokaryotes? So you can see in this diagram, I have the comparison here, but the main one is that, eukaryotes have a membrane-bound nucleus. They also have membrane-bound organelles, and they, for compartmentalization, and they also undergo mitosis. So in comparison, prokaryotes, they do not have a membrane-bound nucleus. They usually have their DNA kind of like just circular within the cell, kind of floating on its own. They don't have membrane-bound organelles, and they undergo binary fission instead. And so you can see here, ribosomes are in the prokaryotic cells because they don't have a membrane surrounding it, so it's not considered an organelle itself, the ribosomes. And then you can see that prokaryotes look a little bit different sometimes because they can sometimes have a cell wall, and they can also sometimes have flagella, usually for mobility if it's needed. But we're going to go into here. So also there are, different types of prokaryotic cell walls that are also something to know about. So the main differences are between gram-positive bacteria and gram-negative bacteria. So gram-positive have this, you can see in this diagram on the left-hand side, they have this really thick peptidoglycan cell wall. And this, usually causes a really purple color. And so when you're looking at, something under a microscope and it's a prokaryotic cell and it's a bacteria, but it looks purple, that's a good indication that it's a gram-positive bacteria.